Hello, everyone. It's good to see you.
Hi, it's me.
Hi.
You don't know who me is?
I know who you are, Vicki. I recognize your voice now.
Well, thank you for all your help.
Oh, you're welcome. You're very
And my printer is screwed up, and I'm having more computer anxiety than anything else right now.
That's the worst kind of anxiety.
I just got off the phone with Staples. I had to give them my computer last week for a couple of days, which started this whole spiral effect of being behind in my homework.
Oh, I can only imagine. It's
now
so stressful.
It's very stressful.
We are completely dependent upon our computers.
Exactly. And I don't know how to use mine half the time.
I know. It's not a good situation.
I apologize for my lateness.
No problem. No problem. I understand. It happens sometimes. All right, ladies, if you would just type your name or your nickname in the chat for me. That's my record of your attendance today. And I'm sorry we missed each other last time, but we had a little windstorm and things got a little dicey power-wise. And Wi-Fi went out as well, even when the power came back, at least where I am.
It's not even letting me write my name.
Oh.
I'm having...
Oh, no.
I'm here. Oh, there it is.
You're here. Yeah, so so we didn't get to see each other last time, but that's okay. So it looks like just about everybody's here. So we're going to shift our scheduling around a little bit. Today we were going to start talking about protein catabolism part 2. But instead what we'll do is we will discuss the first part of the lab which we missed. So today we will begin with protein catabolism part 1. It's not a particularly long lab, so we may get a chance to start into the part 2, but we'll see. And we will just go forward from there. And then next week, we'll just keep moving forward from there. So, all right. I did want to remind everybody that it is March 31st and your final draft is due today of the pathogen project. So I've already received a few of them. Thank you. And you'll want to try to get that to me by midnight tonight, or send me a message and let me know if you can't, why you can't. Okay. All right. Very good. So does anybody have any questions about anything that you've been looking at for lecture? We are into some, heavy lecture material now. We're into DNA replication and transcription and translation, and those are difficult topics. And while I know you have all had biology, I also recognize that the details of processes like this are they're not easy to recall.
Well, just a little heads up, I am very confused and I'm trying to figure this all out. And I'm having a very hard time.
This is a difficult subject. Genetics in general is difficult. And these are processes that are very detailed, and you're asked to not only learn a lot of detail, but you're asked to visualize these processes occurring inside of a cell. And especially if you're far away from the first time you ever learned this, if you ever learned it, because of course we all come from different backgrounds. It's a difficult subject. And those processes, DNA replication and what we call gene expression, which is transcription followed by translation, those processes are key to anything and everything that has to do with the workings in a cell. So the reason we learn them and relearn them and relearn them in multiple classes is because of how important they are. But they don't get easy. They certainly don't get easy as we learn them. So, so that's why it's important while you watch these lecture videos and you take notes and things, if there's something in there that doesn't make sense to you, jot it down and either bring that question to lab or send me a message so we can talk about it. You know, I make these lecture videos for you, but because we're not sitting in front of each other, I don't know if I'm saying something that is not landing with you.
Well,
the only
I do have a question.
Yeah.
And this might help explain a lot, but I'm missing like a definition of a word.
Oh, okay.
Understand. Oh, I can't even find it now. It's like a major part of the process.
Okay. See if you can find it.
Starts with a P, the big long one. Shit, now I can't find it.
I'm trying to think what it might be.
The oxidative phosphorylation.
Very good. That's a mouthful, isn't it?
Yeah, I don't understand. I mean, there's
It's a great question.
phosphorylation.
So remember, it's always helpful for me, I know, to sort of translate some of these big words that we put together and name things. So you know, as you guys know now, I like to translate words to help us better understand them. So that word oxidative just refers to a type of chemical reaction called an oxidation reduction reaction. And we've talked about this. If we call it oxidative, we're saying that electrons are being lost from one molecule and gained by another. And in lecture, we talk about how glucose is getting oxidized through multiple steps, right? So the glucose gets oxidized, something else is getting reduced. Then that thing gets oxidized and the next thing gets reduced. And then that thing gets oxidized and the next thing gets reduced. It's all about losing and gaining electrons. That's all oxidative means.
All right. So oxidative is movement of electrons.
And oxidation reduction. And remember, those two things always go together. So oxidative phosphorylation, that word phosphorylation just means adding a phosphate group to something. And phosphate groups are phosphorus molecules surrounded by oxygens. That's all a phosphate group is. Now, the thing that's getting phosphorylated or having a phosphate group added to it during oxidative phosphorylation is this molecule called ADP.
Right.
If you add a phosphate group to it, it becomes ATP. Right. So oxidative phosphorylation is the kind of phosphorylation that occurs during cellular respiration at the very end. After the electrons have come down the electron transport chain, the energy is harvested from them. And as you saw in lecture, that energy gets used to do something. And the something that gets done is a hydrogen gradient gets produced. Hydrogen ions get moved, get pumped out of the cell till you have this big gradient. You have this big difference in concentration between the outside and the inside of the cell. That gradient has potential energy in it. And what the cell then does is it allows that gradient to resolve, to go away by passing the hydrogen ions through a molecule called ATP synthase. It's an enzyme. It sits in the cell membrane. And this is the enzyme that's doing the phosphorylation. It's adding the phosphate to the ADP. So electrons, if you were to put it into the most basic summary, during cellular respiration and oxidative phosphorylation, all the electrons are being released from glucose. Glucose is broken apart until there's nothing left but carbon dioxide. And all those electrons are going to get fed down the electron transport chain, and their energy, anyway, is going to be taken from them. It's going to be harvested from them. The electrons are received at the end of the electron transport chain by some molecule that gets reduced. Glucose is oxidized. That final electron acceptor is reduced. Might be oxygen, might be nitrate, sulfate, carbonate, depends. Now, meanwhile, we've harvested all this energy from those electrons that came out of the glucose. What are we going to do with it? We build a hydrogen ion gradient, and then we allow all those hydrogen ions to flow back into the cell through, literally through, a molecule called ATP synthase. As they pass through the center of that molecule, through a tunnel in the center of that molecule, they literally cause that thing to spin under the membrane. And as it spins, it's generating energy, or it's expending energy. And that energy is what's phosphorylating and creating the ATP. It's a series of reactions. We're harvesting all the energy up from glucose, and we're converting it into ATP, is essentially what's happening. Remember what the first law tells us. You can't create energy, you can't destroy energy, but you can certainly alter its form. So we often in cells see a chemical, all the energy out of a chemical taken out and converted into something else. All that energy that's in that little glucose molecule. And remember, we say by energy, we mean the electrons, because that's where the energy is. All those electrons are going to be used to build ATP. Oxidative phosphorylation is very efficient. You're going to get 30 plus ATP molecules from one glucose. That's a lot of ATP. Substrate level phosphorylation is very inefficient. You only get one ATP every time the enzyme makes a product. You get one ATP piggybacked onto that. So two phosphorylation mechanisms, two ways to add a phosphate group onto ADP. Substrate level, not a good way to do it. And oxidative, much better way to do it. So it's much more efficient to do cellular respiration and oxidative phosphorylation. But you can still get ATP through fermentation. And some microbes only do fermentation. They just don't make as much ATP because they just don't need as much ATP. But you and I, we could never survive through fermentation. We couldn't survive it because our body needs so much energy all the time, so much ATP all the time, that we have to do cellular respiration as our primary pathway. Even when you're just laying on your bed, not doing a thing but breathing, you're still burning energy, right? Your basal metabolic rate is something like 800, approximately 800 kilocalories per day. You just to exist, Your cells need energy all the time just to exist. All right. I hope that helped a little bit. It's a lot of terminology.
Yeah.
A lot of terminology, for some of us, brand new terminology. And it's a constant battle as you're reading through all this complex terminology or listening to this complex terminology to keep trying to figure out exactly what it's saying.
That's why my quiz ended after question 1.
It's just, it's, believe me, I understand.
So I have another question.
Yeah, please.
Are the dates for everything that's due going to be the same? Somebody just asked.
So the lecture quizzes this week will keep right as they are.
Okay.
What I'm going to have to do with the lab questions is we'll see where we end up today. And then I will adjust the lab questions for the week.
What about the
Reflect that.
exam?
Oh, for next week. So the next, let me pull this up for us so we can all take a look. I did pull it out.
I'm just, my head is swimming.
I feel the same way, I have to say. We're at that point in the semester where it's all just a lot, right?
Mm-hmm.
All right.
Plus life.
Yeah, that's right. All right, hold on just a sec. See if I can pull this up. I don't think it's gonna let me. Come on, Zoom. All right, it's not gonna let me. All right, so next week you have your next lecture exam. So the lecture material is gonna stay right on schedule. The laboratory material is gonna change a little bit. It's gonna get behind a little bit. Now I think we'll still be okay because if you look at our syllabus, I think there's a little wiggle room in there for us to finish before the lab practical exam, the final lab practical, which is scheduled on your syllabus on the tentative schedule. But we'll have to see. We may have to bump the final lab practical one day, but we'll see. Okay. All right. And I will make an adjustment to the lab questions for this week. I'll go in and I'll make an adjustment in terms of which questions to answer. Okay. Or I may even postpone it. I may try that.
So the test is going to be on metabolism and genetics.
I believe those are the two topics. I think those are the only two topics. Let me double check. That's right. So metabolism, which is enzymes and energy, cellular respiration, oxidative phosphorylation. That's your metabolism material. And then the genetics is what you're doing this week, which is DNA replication and mutations, and transcription and translation, which is what we also call gene expression. And just like all the other lecture exams, your exam next week, you'll be able to have your lecture notes with you. So the key thing this week is going to be to make sure that you understand everything and you've got it all in your notes. And again, you know, as questions come up towards the end of the week, don't just hold on to them, send them to me. Okay? Send them to me and we'll get it straightened out for you. All right, very good. So this week in laboratory land, we are leaving carbohydrates behind and we're looking at protein catabolism to be precise. And one of the very first things we need to know is that proteins are used very differently by cells than carbohydrates are. Carbohydrates are the macromolecule that cells use in order to make ATP. They can do it through respiration, or they can do it through fermentation. They can do it through oxidative metabolism or fermentative metabolism, but they're using carbohydrate to do it. Cells use protein for something very different. The most common thing that cells will do with a protein is break it apart to get at the amino acids. Now, this is for microbial cells, bacteria, fungi, and so on, as well as our cells. That protein that you eat in your diet that gets digested, the first thing that happens is the protein gets unfolded. Then you've got a nice long chain of amino acids, and then the peptide bonds between the amino acids get broken, and each amino acid gets released. The most common thing that a cell will do with protein is get the amino acids out. Now it has a bunch of new amino acids that it can just make another protein with. Cells are constantly making protein through gene expression, right? Transcription and translation. So having an abundant supply of amino acids is important. So rather than always spending time and energy building amino acids from scratch, cells will instead break down protein, nutrient protein, to get some amino acids. So that's the number one thing that microbes are doing when they are fed protein. They're breaking the protein down into individual amino acids. Remember, some proteins are enormous. Some proteins have thousands of amino acids in them. So it's a great source of amino acids. Now, there is something else that a microbe could do with protein, and that is break it apart into the individual amino acids and then break those amino acids apart. Now, why would it do that? The most common reason for a cell to break down an amino acid is to get some carbon. Remember, we on planet Earth, we are organic life forms, so we're all built around carbon. And carbon is the basis of all biologically relevant molecules. So cells need a source of carbon all the time because some molecules are built from scratch inside of a cell. Sometimes if a microbe is running low in carbon, what it will need is some molecule that it can dismantle and break apart to get the carbon out. And amino acids can be used for that purpose, as well as other nutrients. So most of the time, a microbe is going to digest a protein down, break a protein apart, and keep the amino acids whole, because it can use those amino acids to build more protein. But some microbes also have the ability to take some of those amino acids and break them down to get at the carbon. All right. Today, we're going to begin by looking at that first thing. We're going to look at some tests we can run to test whether a microbe has the ability to break certain proteins into constituent amino acids, lots of amino acids. I'm going to go ahead and pull up our slides. All right. So you should be seeing protein catabolism part 1 on your screen. The first thing we'll do is we'll just quickly review some of the protein terminology because it's very easy to get those terms confused. I've just summarized for us the most common reason that bacteria catabolize protein, again, to get the amino acids. And then we'll look at 2 tests related to this. The first one is called the gelatin hydrolysis test. We'll talk about what that media looks like and how you perform the test and how you interpret the test. And then we're going to look at another one that's related to protein called the Urease test. Again, what the media looks like, how you perform this test, and how to interpret it. But first, let's review this terminology. This term protein means something very specific. Now, I am guilty of using the term protein when I should really be using the term polypeptide. I do that all the time. Because a polypeptide and a protein, they're close, but they're not exactly the same. A polypeptide is essentially an unfolded protein. The protein is the polypeptide all folded up. So let's start with this term, peptide. A peptide is a short chain of amino acids. They are attached to each other through covalent bonds called peptide bonds, each amino acid to the next one. And if there are a small number of them in this chain, and by small I mean tens to hundreds of amino acids, we call that a peptide. If there are more amino acids in this chain, we call it a polypeptide. If there are hundreds to thousands of amino acids in this chain, we call that a polypeptide. And finally, when a polypeptide is processed and folded up by a cell, it becomes a protein. So a protein is a folded three-dimensional structure. Every single protein has a unique sequence of amino acids in it. In other words, the order that the amino acids are in that long chain is unique for every protein. Therefore, every protein has a unique three-dimensional shape because it's the order that those amino acids are in, it's the sequence that they're in that's going to determine how that thing folds up. Remember, we talked about polar and non-polar. We talked about hydrogen bonding and van der Waals interactions. The way that amino acids are attracted to each other or repelled from each other is going to determine how that long polypeptide strand folds up and becomes a protein, a three-dimensional molecule. All right. So at its most basic form, protein is made out of amino acids. There are 20 amino acids that occur in nature. And those 20 combine to make all of the different proteins that you can find in nature, in cells and living things. Living things, living cells, like microbial cells, also can break apart protein. They can use protein as a nutrient. And the most common reason that they would do that, particularly in bacteria, is to get at those amino acids. So they will catabolize a protein, they will break it apart in order to get amino acids so they can build a new protein that's anabolic. They catabolize to get the amino acids, and then they use an anabolic process to build new protein. And as I stated earlier, it's also possible when a bacterium gets amino acids out of a protein for that bacterium to break apart the amino acids. And that's what we'll talk about in the second part of the lab. So we're going to look at the ability of several bacteria to catabolize a very particular protein called gelatin. Now, you're all familiar with gelatin if you've ever eaten Jell-O or you've ever had any other type of food item that has a gelatinous texture to it. Gelatin is a very common food ingredient. It's actually part of the connective tissue that comes out of animals. So when animals are harvested for beef and pork and chicken and things like that, one of the products you can get out of an animal carcass is gelatin. Now, gelatin is a protein, and it's composed of, again, a variety of amino acids, all bound together in a long chain and then folded up. What this test is going to look for is the ability of certain bacteria to break that gelatin down back into its amino acids. So we're going to perform what's called the gelatin hydrolysis test. Remember, hydrolysis just means using water to break something down. We've seen the starch hydrolysis test. Now we're going to see the gelatin hydrolysis test. Remember, the enzymes that are doing these hydrolysis reactions, these are exoenzymes. So the bacterium is secreting these out into the environment. The gelatin, in other words, is out in the environment. And compared to that little bacterial cell, gelatin is pretty big. It's a big molecule. Now, if the microbe can break the gelatin down, it can absorb or take in those amino acids. So some bacteria make an exoenzyme called gelatinase. There are actually several gelatinases. It's a family of enzymes. Some bacteria can make gelatinases and secrete them out into the environment. And if there's gelatin around, those enzymes are going to break the gelatin down. Now, the other test we're going to look at today is the urease test. And this is not a test that looks at how a protein is being broken down into amino acids. It's just looking at whether a protein is being broken down into amino acids. And here's how it works. When a protein gets catabolized to set loose all of those amino acids, a waste product is produced. And this waste product is called urea, or urea is the other way you pronounce that word. This is a waste product, all right? This is not an amino acid. This is nothing that bacterial cell is going to use as a nutrient. It's waste, okay? Urea. Now, some bacteria make an enzyme that can break down urea, more catabolism. You can imagine that if you're catabolizing protein and you're creating a lot of this waste around you, a lot of this urea waste, it would be really nice to be able to break that waste down too, so it's not so toxic in your environment. The enzyme that can break down urea is called urease. Urease is another of these exoenzymes. It gets secreted. So again, if a microbe is secreting lots of gelatinase and it's breaking down proteins, or maybe it's secreting another enzyme to break down another kind of protein, but it's doing that, it's breaking down or catabolizing protein in its environment, a lot of urea is going to get generated. So we can also ask, well, does that microbe, can that microbe also break down that urea? Does it have the enzyme necessary for that? And that enzyme is called urease. Now, when a microbe secretes urease, the urea is going to be converted into ammonia. And again, this urease test is going to show us if that's happening. Humans also generate urea when we break down protein for amino acids. And urea is toxic for us too. In fact, we monitor it. We monitor blood urea nitrogen, or BUN, as a measure of how well the kidneys are working, because the kidneys are going to help get rid of urea. When urea gets broken down and ammonia gets produced, it makes your urine have a certain smell, right? It's the ammonia. It's the breakdown of urea in the urine that gives it that smell, that ammonia smell. So it's happening in some bacteria. It's happening in us and other animals. We can test for it. We can test for it in our microbes to see if they're able to do it. Okay. So this first test, this gelatin hydrolysis test, involves what we call nutrient gelatin medium. This is essentially a nutrient broth that we're going to modify. All right. So the ingredient list is here, and it's very limited. Okay. There is gelatin in here, of course, because we want to see if the bacteria can break it down. There's also some gelatin that's been partially digested already. That's going to help because gelatin is such a big protein. It takes a certain amount of time for it to break down. And so by also putting partially digested gelatin in there, we kind of help the bacteria along a little bit. If they're able to secrete gelatinase, they'll be able to break down that partially digested gelatin a little bit quicker. You'll notice that there's also something in here called beef extract. This is one of those complex ingredients that we've talked about. Beef extract is something that comes from cattle when they are slaughtered. It is a complex ingredient. Sometimes when people see the word beef, they think that that means skeletal muscle. But that's actually not what beef means from a nutritional standpoint. Beef is actually a complex mix of all of the kinds of tissues that you would find in and around the skeletal muscle. So yes, there's protein in it. There's also carbohydrate in it. There's fat in it. There are minerals in it. There are vitamins in it. It's very complex, beef extract. Now, one thing you'll notice about this very short list of ingredients is there's no agar in here. How in the world can we grow bacteria on this medium if there's no agar in it to make it solid? Well, the answer is the gelatin will make it solid. As you know, if you've ever had Jell-O, gelatin makes a nice solid, it's soft, but it's solid material. So the gelatin is doing the work here that agar would normally do to solidify the medium. Now, here's what you need to know about gelatin, which is different from agar. This medium, nutrient gelatin medium, is going to be a liquid above the temperature of 25 degrees Celsius. Now, if you're not familiar, room temperature is 22 degrees Celsius. So at room temperature, gelatin is solid. Again, think about a bowl of Jell-O. It's going to be solid in the refrigerator. It's going to be solid at room temperature. But if it gets much warmer than room temperature, it's going to melt, right? It's going to liquefy. Now, when gelatin is broken down because a microbe is secreting gelatinase, the gelatin is going to go away. It's going to be gone. And all that's going to be left are the amino acids. So at that point, the medium will become a liquid, even at room temperature. And that's how we're going to know how to read this test. We're going to take this solid gelatinous medium. We're going to inoculate it with bacteria. We're going to allow it to incubate. If we come back after incubation and it's still solid, that tells us that the gelatin is still there. So the bacterium must not have broken it down. It does not have the gelatinase enzymes to do that. If we come back after inoculation and incubation and the tube is full of a liquid at room temperature, that tells us that those cells did break down the gelatin and released all of the amino acids. Now, there's a key thing to know here, and that's this. The incubator that we normally use is at about 35 to 37 degrees. So you can't incubate this test in the incubator. You can't put it into a hot incubator to let the cells grow, because everything's going to turn liquid in there in terms of this test. So instead, what we do is we inoculate the test and we put it into the incubator turned off. We're going to put the tubes into the incubator, but we're going to shut the incubator off and allow it to come to room temperature. It'll still be a nice, dark environment, protected environment for the cells to grow in their tubes, but it won't be warm. It takes a while to run this test. It can take a week to run this test, depending on the microbe. It takes them a while to break down that gelatin. So it can take as long as a week. Sometimes it happens sooner if they have the ability to catabolize the gelatin. Sometimes it happens within a day or two, but you wouldn't want to judge it as being negative until you had waited at least 7 days. If at 7 days that tube is still solid at room temperature, that's a negative test. That organism cannot break down that gelatin.
Can I just ask one question just to clarify? So it's liquid up to 22 where it solidifies.
It's liquid. It's actually liquid until it's solid until 25.
Okay.
So room temperature is around 22. You know, rooms are going to vary a few degrees. But remember too, this is centigrade. So around room temperature, this thing is going to be a solid. And it will not liquefy until it hits 25 C. It'll start to liquefy at 25 C. Okay, that would be a very warm room. That would not be a normal room that you were sitting in. Okay, that might be out in the sunshine.
Gotcha.
Yeah.
Thank you.
So that's why we keep these tubes at room temperature until we evaluate them. Remember, that's one of the beautiful things about agar. One of the reasons we like agar so much is that it stays solid in the incubator, right? That one, agar doesn't melt until about 100 degrees centigrade. So gelatin would not make a good culture medium. It would not make a good thing to solidify culture medium because it would melt in the incubator. Okay, so on this slide, you're looking at 2 of these tubes as they're being evaluated. So these are glass test tubes. This is what the gelatin tube looks like before it's inoculated. Now it's being held on its side right now to read it, but it looks like a deep, doesn't it? It's got solid material in it. It's not agar because it's gelatin instead, but it's this nutrient gelatin medium. And it's solid. Now we put the cells in, we incubate it at room temperature, and then when we're ready to read it, we have to tilt the tube because that's going to tell you whether this stuff has liquefied. You can see that this one has not. This tube has not liquefied, but this one has. I know it looks like a slant right here because it's a still image, but what this is liquid. If you were to roll this tube around in your hands, in your fingers, this is liquid. This is not a slant tube. So gelatinase negative, the gelatin is still present. The medium is solid. those bacteria in there cannot make and secrete gelatinase. They can't break down gelatin into its amino acids. This tube, this is liquid. The gelatin is gone. This is a positive test for gelatinase. These bacteria made and secreted the gelatinase enzymes, and they broke down the gelatin that was in the test medium. If I were to take a little bit of this liquid out and take it into the chemistry lab, it would be full of amino acids, but there would be no gelatin in here anymore. All right. Questions about that? Just as an aside, I'll add in, gelatin is not a horrible protein. You know, there's a reason why mothers and grandmothers feed children gelatin when they're sick. It's a protein. Sometimes people look at gelatin and they think, oh, that's a, oh, it's terrible, blah. But it's a protein. Now, it's not a, high quality protein. There are better proteins. But for the purposes of getting protein into a sick child, Jello's not bad. Jello's not bad at all, because we have the enzymes that are necessary in our gut to break it down and get the amino acids out. I always laugh because they still, I believe, they still use Jello in some situations in the hospital. They'll still feed Jello to people who won't eat anything else because they need to get protein into you if you're sick. So for some people who, can't stomach any other food, a little cup of Jello, that's good. That's protein. All right, we're going to switch gears now and talk about this urease test. Now, this test is also a solid culture medium. I shouldn't call it a culture medium because it's not used just to culture cells. It's used for this test. It is a solid medium, though. It has agar in it. You can see it down here at the bottom of the recipe list. It's a very simple recipe, right? This is a basal medium. There's no complex ingredients in here. There's no beef extract. You can see sodium chloride, monopotassium phosphate.
Yes, love.
I'm sorry, say again.
Oh gosh, I didn't know I was on mute.
Oh, that's okay. That's okay. So salts and minerals. Here's dextrose. That's carbohydrate. That's got glucose in it. Here's peptone. That's the partially digested protein. So a basal medium, basic ingredients that will support the growth of non-fastidious bacteria. Now, we supplement that with urea because we want this urea around because we want to see if they can break it down. Here's phenol red. That's an indicator dye. So we're going to be looking for a color change. And here's the agar that's going to solidify it for us. Now, we're going to look at urease medium in a slant in just a minute, but I do want to mention that you can also make it or buy it as a broth. In my experience, most laboratories use the solid. They use the slant test tube for the urease test, but it is possible to buy urease medium and even to make it as a broth. Now, one of the interesting things about this test medium is that recipe that I just showed you will mix up to be slightly acidic. And it has phenol red in it. Remember, phenol red we saw in a couple of tests so far, including that carbohydrate fermentation medium. Remember, it starts off red in that test, and we look for it to turn yellow with the production of an acid. Well, in this case, it's actually going to start off yellow because the recipe itself makes the medium acidic. Okay, so this time, this medium is going to start yellow in color. Now, what's going to happen if the urea gets broken down is ammonia is going to get produced. And that's going to raise the pH. That's going to turn this media alkaline. And the phenol red is going to make this really nice fuchsia color, bright, bright pink fuchsia. That's going to tell us whether the urea has been broken down. So we're going to take the test medium, we're going to inoculate it with bacteria, and we're going to incubate it for 24 hours, this time in the incubator, in the hot incubator. But here's an important thing to note. Put a star next to this. We're not just going to put the urea test tubes into the incubator and just forget about them and come back the next day and read them. Instead, we're going to read them at intervals. We're going to go back and we're going to check those tubes every hour or two, looking for this color change. It turns out that among the bacteria that secrete urease and break down urea, some of them do it really fast. Some of them, the tube will turn fuchsia or bright pink in about 6 hours. That turns out to be an important thing to know because that is a characteristic that only a small number of bacteria have. So you don't want to leave this tube for 24 hours because by 24 hours, lots of bacteria will have turned the tube pink. So we want to know, is it rapidly positive or is it more slowly positive, 24 hours, or is it negative? So this is urease medium right here in a slant. I call it yellow, but it's really sort of a yellowy orange, almost a peach maybe, or an apricot if I was going to try to be descriptive. So it has phenol red in it. And because it's slightly acidic, it starts off with this yellow orange color. After I inoculate this, after I streak some cells on this diagonal piece of agar, and I incubate those cells, if they can secrete urease, this test medium is going to become alkaline. The pH is going to go up above 7, and this is going to happen. This is a positive urease test. No question, right? There's no confusing this tube and this tube. That is a marked color change. The key thing is to figure out, did it happen quickly or did it happen more slowly? Because that's going to help us identify the bacterium. I put these pictures in just again to show that you can make up a broth for the urease test. This is how it looks before it's inoculated. This is what a positive test is going to look like. Again, fuchsia. And this is what a negative test is going to look like. No color change. So we're literally asking if the bacterium is making and secreting urease, but that means something as well. That means that that organism in its normal life must break down protein into its amino acids because that's how urea gets made. And it makes sense that if you are an organism that catabolizes protein to get to the amino acids, you would also want to be able to make urease, evolutionarily speaking. Because remember, a microbe is subject to its environment. So if it starts secreting the enzymes needed to break down the protein that surrounds it in the environment, so it can absorb all the amino acids, it's very quickly going to be also surrounded by urea. So it makes sense evolutionarily that if you're a microbe that is able to do this, you're going to also want to be able to deal with that urea. You're going to want to be able to secrete something else to break apart that urea so it doesn't kill you as it builds up and up and up and up.
So we don't have urease because we have kidneys, correct?
Well, we do have kidneys, but we also have urease.
Oh, we have urease.
Yeah.
Okay.
The kidneys are going to help get the urea out of the blood. And then the urease is going to break the urea into ammonia.
Gotcha.
Yes, our kidneys are amazing. Poor bacteria, they don't have any kidneys. Okay, now, before we go ahead from here, I want to say a few words about a particular microbe called Helicobacter pylori. This is a digital image of this type of bacteria. You can see it's an elongated cell, and it has several flagella at one end. It's funny, you know, you would be forgiven if you called this a spirillum. It looks like a spirillum, doesn't it? It's a little bit twisted, a little bit helical, but not a lot. Doesn't really look like a spirochete. But remember, spirilla and spirochetes are often pretty long cells. Remember we looked at some images? And this one really isn't a very long cell. So they don't call it a spirillum. They call it a curved rod. Just a little factoid for you. Helicobacter pylori is a curved rod. This organism has taken on a lot of significance in recent years. I'll say the past 30 years, because it was discovered to be the cause of gastrointestinal ulcers in many situations, not all, but many. In the old days, when I was a child, for example, it was believed that gastrointestinal ulcers were the cause or were the result of stress. That if you lived a stressful life and you were constantly bathed in stress hormones, that your gastrointestinal lining could ulcerate. What was discovered, again, a few decades ago now, is that sometimes it's not all about stress, it's about a bacterium. Now, if you think about it, this is an amazing bacterium because it's able to live in some pretty hostile parts of the gastrointestinal tract and cause ulcers. It can burrow in to the gastric mucosa, for example, and cause ulcers. So nowadays, if you have symptoms of an ulcer, a gastric ulcer or a duodenal ulcer, that's in the first part of your intestine, one of the things that the physician is going to be thinking about is Helicobacter. And depending on, you know, whether you want to treat first and then really figure it out, see how you respond to the treatment, depending on the physician, really. Sometimes a physician will get a biopsy. And that tissue, once it's biopsied, we can bring to the microbiology lab and we can look for this organism. And one of the ways that we can tell that physician pretty quickly if we think that patient has Helicobacter pylori is by running a urease test. We can take, for example, the biopsy tissue, and we can use it to streak the urease slant, put that tube into the incubator, and that tube is going to turn pink within 6 hours. So Helicobacter pylori is an organism that is rapidly urease positive. So if you think about it, it's handy because we can report back to that physician very quickly that they may well have Helicobacter. There are other organisms that are rapidly urease positive. But again, in a patient who has symptoms of ulcer, in a patient where the physician thinks this looks like Helicobacter pylori, if you get a rapidly positive urease test, you've got a pretty good reason to believe it's Helicobacter. So physicians will start patients on antibiotics at that point, and hopefully the patient will get better. Now, we don't stop with the urease test. We would never stop with one test. We're going to also use that biopsy specimen to do other tests in the laboratory. We're going to want to look at that thing on a smear. We're going to want to see if it looks like Helicobacter. We're going to want to do all the things we normally do, gram staining and so on. But a rapidly positive urease test is a pretty good confirmation of Helicobacter. Now, I said that there are other organisms that are rapidly urease positive. This is one of the times we can talk about a yeast. Remember when we were talking about the fungi, we said there's a particular genus of yeast called Candida. And there are 2 Candida that can be pathogenic and can cause pretty significant disease in humans. And we can use the urease test to tell them apart, at least, as one of the ways we would tell them apart. So I'm going to go back here. The 2 types of Candida that I'm talking about are Candida albicans, which is one of the most common Candida species that causes disease in humans. Albicans, by the way, means white. So I've got the name right down here at the bottom of the screen for you. Candida albicans got its name because it's the organism that can cause things like thrush. If you've ever seen thrush, which is a fungal growth in the mouth, it grows as white patches, and it's often caused by this Candida albicans organism. There's another.
What was the second one? Sorry.
Oh, that's okay. There's another Candida called Candida neoformans. It's quite a name. That can also cause, again, fungal infections, yeast infections in people. And so it's hard, it's tough to know whether you've got Candida albicans, which is very common, or Candida neoformans, which can be quite pathogenic. One of the ways you can tell them apart is with the urease test, because Candida albicans is going to be urease negative. That's what you can see down at the bottom of the slide here. And Candida neoformans is one of these rapidly urease positive organisms. So again, sometimes, we talk about these tests as a way to identify bacteria, and they are certainly used that way. But sometimes it's also very useful clinically. These tests help us distinguish one species from another in a clinical situation. Just to show you the rest of this slide, this is a scanning electron image of some gastrointestinal mucosa. And you can see them. You can see the little cells, the little curved rods all over that surface. Helicobacter. This one is taken with a light microscope. This is a gland in the either the stomach or the intestine. The white part is where the inside cavity of the gland is. And see this little fella right there? It's hard to see, but that's one of these curved rods, and it has lots of flagella coming off of it. This is a Helicobacter. And then this one over here is just to show us that we would, if you were to gram stain this thing, it would come up as gram negative. All right. So for our experiment with our tests today, we're going to use 5 different organisms. We'll use 2 organisms for the gelatin hydrolysis test, and we'll use 3 for the urease test. You've seen most of these organisms before. Staphylococcus aureus is on the list. Staphylococcus epidermidis. Remember, both of those are gram-positive cocci. They're both found on the skin. They could be found in the back of the throat or up the nose. For some people, they carry these organisms. In other words, it's just part of their microbiome. Some strains are pathogenic. We've talked about this before. So we will compare those 2 using the gelatin hydrolysis test. For the urease test, we're going to look at E. coli, Klebsiella Pneumoniae, which we've seen before, and a new one for us, which is Proteus mirabilis. All three of these organisms are gram-negative rods, and all three of them can be found in the gastrointestinal tract. E. coli, as you know, is a coliform. That means it ferments lactose. Klebsiella pneumoniae is also a coliform. We said last time that Klebsiella is capable of causing disease in other parts of the body. Remember, it can cause things like pneumonia, hence the name Pneumoniae for the species. I do want to say a couple words about Proteus. So Proteus can be found in the intestine. It tends to live down in the colon. And there are strains of Proteus that are not harmful to us at all. There are other strains that are pathogenic. You can also find Proteus out in the environment. Now, pathogenic strains of this organism cause all kinds of different problems. It is a cause of urinary tract infection, that's a UTI. You can get respiratory infections that are caused by Proteus mirabilis. And one of the big problems this organism causes is infections in burn patients and sepsis in highly compromised patients. Proteus mirabilis is what we call an opportunistic pathogen. These are pathogens that don't always cause disease. In healthy people who have fully functioning immune systems, they often are not capable of infecting tissue and causing disease because the immune system gets them too quickly. But in other cases, when the patient is severely compromised, particularly, as it turns out, in patients who have lost part of their skin through burns. Remember, your skin is an important barrier to your body. So if you've been burned significantly, you've lost that barrier. And opportunistic pathogens will take advantage of that. So Proteus is often found growing in burn patients. You want to try to prevent that as much as you can if you're treating a burn patient, because Proteus can kill a burn patient if the infection gets out of control. So another interesting organism for us to look at.
If you're looking for a root word as a mnemonic, Proteus and protean means change.
That's exactly right. So if you think about it, I can't say this for sure because I've never studied the derivation of why they chose Proteus for this organism, but it does seem to change in its ability to cause disease. It seems to sort of linger, not causing any problems until the patient becomes particularly compromised. So that same burn patient before they were burned could have Proteus all over them and probably not get sick. But because they've lost that barrier, because they've been burned and they've lost that barrier, the Proteus takes advantage and establishes an infection. Good point. Thank you for that. All right, so here's our gelatin hydrolysis test. These tubes have been inoculated separately with Staphylococcus aureus and Staphylococcus epidermidis. The tubes have been left to incubate at room temperature for upwards of 7 days. And then when we read them, we have to tilt them so we can tell whether or not the media has liquefied. You can see that the S. epidermidis tube has stayed solid. Staph aureus has liquefied. We've looked at these two organisms before, haven't we? These two organisms can be hard to tell apart from each other. They're both staphylococci. They're both going to gram stain the same way. They can both grow on a multitude of media. We looked at MSA, we looked at mannitol salt agar as a way to tell them apart. Remember, S. aureus turns the media yellow. S. epidermidis doesn't. Now we have a second test that can tell them apart. This gelatin hydrolysis test can tell them apart because S. epidermidis will be gelatinase negative, and Staph aureus will be gelatinase positive. These are the results of the urea test. You've got 4 tubes here labeled A to D. Again, these are slants, agar slants with urea medium in them, urease medium, I should call it. This tube over here on the left, this is your uninoculated tube. Remember, this is a medium that starts off sort of a yellowy orange color because it's slightly acidic, and there's phenol red in here. This is Proteus mirabilis growing on this slant, and boy, oh boy, is that a positive. This is an organism that will be rapidly urease positive, Proteus mirabilis. So if I inoculated a tube with Proteus and I put it in the incubator and came back about 6 hours later, the whole tube would be bright pink. Very, very rapidly positive. Compare that to Klebsiella. Klebsiella Pneumoniae, if you took it out at about 6 hours, would look like this. This one is not rapidly positive. We've seen something like this before, haven't we? When you get half of a color change, half of the test medium has stayed yellow, the rest of it has gone pink. This just means that Klebsiella Pneumoniae is not rapidly positive, it's more slowly positive. And if we were to put this tube back in the incubator and let it cook for some more time, 24 hours, the whole tube would be pink. The whole tube would be pink by 24 hours. That's what I mean when I say you've got to check these tubes at intervals. If I came back the next day, I would have 2 fully pink tubes. It would be hard to gather any information from that. When I check them at 6 hours, I see a big difference between those organisms. Here's E. coli over here on the right. Good old E. coli. E. coli is urease negative. So that medium is going to essentially stay the acidic color, the yellow color. Now, sometimes I'll have students ask, how come the E. coli tube is more yellow than the uninoculated tube? And that's a great question. The reason has to do with the phenol red again. Remember, phenol red becomes yellow as pH becomes acidic. So it starts off as that sort of orangey yellow because the pH is acidic, but it's not very acidic. It starts off at like 5.5. E. coli, as it's growing in that tube, because it will grow in that tube, there's a basal medium in there. There's a nutrient medium in there. E. coli is going to grow, but it can't break down the urea because it doesn't have the enzyme to do that. As E. coli grows, we know it's going to use that carbohydrate, that dextrose, to make ATP. And as it does that, it's going to create an acid. So the reason that tube turns even more yellow for E. coli is because it's metabolizing as it grows and it's producing acid. It's just not breaking down the urea. If it broke down the urea, the pH would have gone way up over 7 and the medium would have turned pink. I want to say one more thing about this picture that's important. Anytime you run a biochemical test like this, where you are using a special test medium and it has an indicator dye in it, you always run an uninoculated, see if I can speak, an uninoculated control tube. And what I mean by run is I'm going to put this tube through the same process that I put these tubes through. This tube doesn't have any cells in it. I haven't done anything to this tube, but I'm still going to put it in the incubator along with these. I'm still going to let it incubate for 24 hours. I'm still going to look at it the next day. I'm using it as a control. Now, what can it tell me? It can tell me if there's a problem with my culture media. We haven't really talked about this, but indicator dyes are wonderful. They are wonderful. We've seen them again and again in here. They change color for us and they make us able to see what's happening in the tube. But indicator dyes get old, and indicator dyes can malfunction as they get old. So part of what I'm doing with that uninoculated tube is I'm making sure that there's nothing wrong with my test media. Let me close that. In other words, let's say I put that uninoculated tube into the incubator with all the other tubes, and then I came back the next day and I looked at that uninoculated tube and it turned pink. I've got a big problem, don't I? I didn't put any cells in that tube. How in the world did it turn pink? That would be a sign that there's something wrong with the test medium. A cell didn't break down that urea, but somehow that test medium became very alkaline overnight. That would tell me something important. That would tell me not to trust whatever the other tubes were showing. I would have to toss everything at that point and run the whole test over again. Make some fresh medium, make some new tubes, inoculate them again. I would have to do the whole thing over again. Controls are there for a reason. They're there for comparison purposes. Yes. And one of the things that we are comparing is how they look before and after the incubation period, before and after the test is run. We expect that uninoculated control tube to stay exactly the same. And if it doesn't, that's an indication there's something wrong with our test medium. So we always run controls. Always, 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 always. It's the safest thing to do. All right. So when we get together next time, next week, we're going to move on to catabolism part 2. I'm not going to jump into it right now. I'm going to let you guys go. So what that means is I'm making an executive decision on the fly. I'm going to postpone the homework for this. All right. So it's not due Sunday at midnight. I'm going to leave the assignment the way it is, and I'm going to reassign it for next week. Okay. After you've had the second part of this lab. Okay. So you have a little bit less load this week. You don't have to do any lab homework this week. So what I want you to do, because you do have a lecture exam coming up next week, what I want you to do is really go through your lecture notes for metabolism and genetics and make sure that you understand those very difficult topics. Get those quizzes done and get your notes in shape for next week. And if any questions come up, let me know. Just because we're not meeting again as a class doesn't mean we can't talk about lecture material. Okay. I think that's all. Does anybody have any questions? I don't see anything in the chat. Anybody have any questions? Questions about what we talked about today? Questions about what's coming up next week? All right. All right, very good. If you have not heard, the vaccination schedule has greatly accelerated in the state of New Hampshire. Hopefully you've all heard this, but we are rapidly approaching the date when people, young people basically over the age of 16 are going to be able to sign up for a vaccine. So be sure to keep your eye on that. I can't tell you the exact date, but it's coming up in April. And it'll be an opportunity, in other words, to sign up, to get a date to get vaccinated. Originally in New Hampshire, it sounded like that was not going to happen until about May 1st. But that has been accelerated now. So they're opening up the scheduling to younger and younger and younger people very quickly. Just so you're aware. All right, everybody, if there are no questions, I'm going to let you go. Okay? Let me know if anything comes up. And we will see you next week.
Thank you.
You're very welcome.